Good afternoon. My name is Serrato Diaz, and today I'm going to talk about mango anthracnose management from field resistant to cold storage solution. This is a collaborative project between USDA ARS STARS in Mayagüez, Puerto Rico, and Dr. Jair Aaron in Martex's farm. As introduction, mango is ranked fifth in total production among fruit crops worldwide. In the United States, mango production is concentrated in Florida, Hawaii, and Puerto Rico. In Puerto Rico, 20,000 tons were produced for 2024. The main consumers of Puerto Rican mango are Europe and the United States. Anthracnose caused by Coleotrichum is the most important post-harvest disease that affects shelf life in mango. Controlling anthracnose is very important since this pathogen can rot mangoes in 8 days of shipment. The figures at the bottom are how the disease looks like on leaves and mango fruits. There are also how the conidia looks like under the microscope and the color and texture of the colony. As I said, this is a collaborative project between USDA ARS and Martex Farm. This project was funded by the National Mango Board, and it has as a main objective to discover possible sources of resistance to mango anthracnose. It has three specific objectives. The first one is to test if low temperatures help in reducing disease severity and prolong shelf life for mango exportations. The second one is to test susceptibility of commercial mango varieties and mango species to mango anthracnose using Coleotrichum inoculations in the laboratory. And the third one is to evaluate resistance to mango anthracnose in a population of crosses between promising mango species and cultivars. For the first objective, test if low temperatures help in reducing disease severity and prolonged shelf life for mango exportations. The Coleotrichum inoculum was obtained, Coleotrichum asianum specifically, was obtained from infected leaves of 18 Mangifera indica varieties planted in Charles Isabella Research Farm. This objective had the following purpose: evaluate if stored temperatures at 11 Celsius can control reduced anthracnose severity. For experiments with temperature, 24 mango fruits of 8 Mangifera species and 7 Mangifera indica varieties were stored at 11 Celsius, simulating an international shipment, and 25 Celsius simulating local market for 15 days, and then transferred to 25 Celsius for 10 days more, simulated arrival at international supermarkets. Anthracnose was evaluated at 15 and 20 days after inoculations. We had 4 treatments. Treatment 1, mango species varieties inoculated with Coleotrichum asianum at 11 Celsius. Treatment 2, mango species varieties inoculated with water only and stored at 11 Celsius. Treatment 3, mango species varieties inoculated with Coleotrichum asianum at 25 Celsius. Treatment 4, mango species varieties inoculated with water only and stored at 25 Celsius. The picture on the right shows the stage of development of mango fruits for inoculations. Mangoes were harvested as regularly Martex does for exportations. The Martex fridge were set up at 11 Celsius degrees. To prevent cross-contamination, parafilm was used to cover plastic containers where the inoculated mangoes were placed. The graph on the left shows the anthracnose severity at 25 days for different temperature treatments. The x-axis represents the temperature treatments, while the y-axis represents the proportion of anthracnose severity. As you can see, the yellow bar, fruits inoculated with Coleotrichum and stored at 11 Celsius had less anthracnose severity than fruits kept at 25 Celsius. That means that treatment at 11 Celsius can reduce anthracnose severity. The graph on the right is a composite graph of anthracnose severity response at 10, 15, 20, and 25 days for after inoculations for 8 Mangifera species and 7 Mangifera indica cultivars. The x-axis shows the time, 10, 15, 20, and 25 days after inoculations, either with water or cholecalciferum. The y-axis shows the anthracnose severity. Blue lines are fruits inoculated with water, while the green lines are anthracnose response for fruits inoculated with cholecalciferum. As is shown in the figure with yellow stars, Kid and Mangifera species 1 had less anthracnose severity over the time. That means that possible Mangifera species 1 could be a new source of resistance. Now let's analyze the commercial varieties Mahachanok, Irwin, and Nandokmai, labeled with red stars. Remember, blue lines are treatments with water, while the green ones are treatment with cholecalciferum. Green light lines are fruits infected with cholecalciferum and stored at 11 Celsius, simulating sea shipment to Europe for 15 days, and then transferred to 25 Celsius for 10 days more, simulating arrival to international supermarkets. The dark green lines are fruits inoculated with cholecalciferum and kept at 25 Celsius all the time. For these varieties, kept mangoes at 11 Celsius for 15 days help to reduce anthracnose severity. However, when the mangoes are transferred to 25 days and kept at that temperature for 10 days more, simulating arrival to international supermarkets, the fungus continues growing. And in some cases, as you can see, graph for Mahachanok and Nandokmai, the anthracnose severity reaches the 100% of infection at 25 days. For the second objective, test the susceptibility of commercial mango varieties and mango species to mango anthracnose using cholecalciferum inoculations in the laboratory. 8 species of mango, Mangifera casturi, Laligigua, Laurina odorata, Rubopretella, Sylvatica, Laurina, and Ceylanica, and 7 Mangifera indica cultivars, Irwin, Kensington Pride, Kid, Mahachanok, Nandokmai, Tutapuri, and Sonny, were inoculated with cholecalciferum asianum. And we use the scale to 0 to 6 to evaluate resistance, as is shown in the figure. The graph on the left shows the average of anthracnose severity for species of Mangifera and Mangifera indica cultivars at 25 days after inoculations. X-axis shows the Mangifera species or cultivars in indica, and the y-axis is the anthracnose severity. As is shown in the figure, Mangifera species 1, Kid, and Sonny had less anthracnose severity than other species and cultivar used in the experiments. Using the scale mentioned in the previous slide, Mangifera species 1 and Kid were tolerant to anthracnose severity at 25 days after inoculations. The figure on the right shows how the fruit looks like 15 days after inoculations with cholecalciferum asianum. At the top is showing the anthracnose severity for Mangifera indica cultivars, and at the bottom for Mangifera species. To know if the anthracnose severity was caused by cholecalciferum asianum and not for other fungi from field, Koch postulates were conducted. Diseased tissue of mango fruits from the experiments were plated on potato dextrose agar, PDA, to promote development of fungi. Purified isolates of each treatment and varieties or species of Mangifera were grown in liquid media for DNA extractions. Sequences of 5 nuclear genes were used to identify isolates recovered in the pathogenicity test. A phylogenetic tree, figure on the right, with these sequences was constructed to compare molecularly the original isolate inoculated in the experiments with those recovered from the pathogenicity test. As is shown in the phylogeny, the original isolate that is represented with a circle clustered with those recovered from pathogenicity tests represented with triangles. For the third objective of this project, evaluate resistance to mango anthracnose in a population of crosses between promising mango species cultivars. One tree of a kit was planted close to one tree of Mangifera species 1. A net cage with a beehive inside the cage was used for the crosses. 67 individuals from this cross were selected based on morphological characters such as size and color of the fruits. This population was created in Martek's farm. These fruits from this population were planted in plastic bags, and F1 scions were grafted into Masyahuesano rootstock. To date, 44 crosses between kit and Mangifera species 1 have been evaluated for anthracnose resistance, and 21 are remaining for the summer 2025. The data that I am gonna talk about in the following slide is for these 44 crosses. This graph shows anthracnose severity at 8 days after inoculations with Coletrotricum asianum for 44 crosses of Mangifera indica kit with Mangifera species 1. We set up in this graph a threshold of 30% of anthracnose. As is shown in this figure, 27 crosses were less than 30% of anthracnose severity after 8 days of inoculations. This graph shows anthracnose severity at 15 days after inoculations. As is shown in this figure, the number of crosses that were maintained with anthracnose severity less than 30% were only 11. This graph shows crosses that had anthracnose severity less than 30%. In this graph, only 2 crosses, the cross 10 and 51, had anthracnose severity less than 30%. For 25 days after inoculation with Coletrotricum asianum, only 1 cross, the KC10, had less anthracnose severity than 30%. In average of all anthracnose severity for days 8 to 25, 12 crosses were moderately tolerant, 1 cross KC51 was tolerant, and just 1 cross KC10 was strongly tolerant. In summary, mangoes inoculated with Coletrotricum asianum and stored at 11 Celsius for 15 days exhibited lower anthracnose severity compared than those stored at 25 Celsius. However, the presence of this pathogen in a susceptible mango variety, whether the infection occurs in the field, packing facility, or at the port of departures, indicate that Coletrotricum asianum can survive sea shipment conditions at 11 Celsius. The Mangifera species 1 exhibited significantly lower anthracnose severity at 25 days after inoculations compared to others evaluated species and cultivars. This suggests that Mangifera species 1 represents a promising new sources of resistance to mango anthracnose, performing as well or as better than kit, a cultivar already recognized for its resistance. The wild Mangifera species 1, identified as a promising new source of resistance to anthracnose, was successfully crossed with the tolerant commercial cultivar kit to generate the F1 population of 66 individuals. Anthracnose resistance was evaluated at 8, 15, 20, and 25 after inoculation. At 8 days after inoculation, 27 individuals exhibited less than 30% of anthracnose severity, indicating a relatively high level of early resistance. However, the number of resistant individuals declined over the time, with 11 at 15 days, 2 at 20 days, and only 1 individual remained resistant at 25 days after inoculations. This progressive loss of resistance suggests that the trait is governed by multiple genes. However, further genetic analysis such as QTL mapping could identify the underlying loci and guide breeding for durable resistance. For the next steps, anthracnose have been evaluated in 44 kit by Mangifera species 1 crosses. Only 21 crosses are remaining to evaluate in this summer of 2025. DNA of leaf of F1 population were sent to USDA ARS Miami Station for molecular identification of resistant genes and QTLs. I would like to thank to the field staff in Martex Farm to provide us a mango to conduct the experiments, to Mango and National Board to provide funds to do this research, and the ORISE participants, Adriana Figueroa and Valeria Perez, for all the help in the lab. Thank you for your attention, and I'd be happy to answer the question in this moment.
Thank you, everyone. So as I mentioned, Dr. Serrato is online with us to answer any questions. What I'm going to do right now is please ask her to turn on her camera. And again, everyone, feel free to ask any questions. Do we have any here in the room? Okay, so here we have a question from online, Dr. Serrato. And it mentions what can be done to reduce that anthracnose in the field, right? Do you reduce, and post handling as well? Do you reduce humidity? What are some sanitation practices that you would recommend?
Well, actually, we didn't perform anthracnose inoculations in the field. Just was a post-harvest screening. And actually, I think Dr. Jair Aaron applied some fungicides in there, but this is the point of all this research to, to reduce fungicides applications in the field. But right now, we cannot control the, the humidity. And actually, in my experiments, I increase the, the humidity to get the disease for all the, the screening of resistance.
Perfect. Any additional questions? Okay, thank you so much, Doctora. Again, if you have any additional questions that come up as time progresses, Dr. Serrato's email is on the agenda and you're free to send her an email about that, okay?
Thank you.
Thank you so much.